So let's jump into it and learn more about the importance of cerebrospinal fluid analysis and cell counts. Cerebrospinal fluid, often abbreviated as CSF, is the fluid that bathes the brain and spinal cord. It holds crucial information for informing the diagnosis of infection, subarachnoid hemorrhage, malignancies like leukemia and lymphoma, as well as demyelinating diseases like Guillain-Barré. CSF collection is an invasive procedure requiring a lumbar puncture or spinal tap, where 4 tubes are carefully filled to be sent to the clinical laboratory for different testing purposes. The different tubes may be designated for different tests. In this example, cell counts, which will be the focus of our discussion, are performed on tube 3. This varies from lab to lab and even patient to patient. CSF cell counts are considered an urgent test primarily because cells degrade rapidly in the CSF due to its low protein concentration compared to that of plasma. Cell counts are critical in the analysis of CSF and are generally performed in either the hematology lab or the microbiology lab, depending on where you are in the world. A total cell count is performed to quantify the total number of red blood cells or nucleated cells or white cells in the CSF. Normal CSF does not have any red blood cells and has very few white blood cells. With age, the normal reference range for white blood cells actually decreases. A differential cell count can be performed to quantify the types of white cells, and the ratio of white blood cell types provides important information to the clinician as to what might be going on with a patient. As mentioned in the previous slide, CSF should not have red blood cells in it, so when red blood cells are present, it warrants for further investigation. Red blood cells in CSF may be indicative of a hemorrhage where there's bleeding into the CSF, or maybe a traumatic tap from the collection needle puncturing a blood vessel on the way in. Previously, I mentioned that multiple tubes of CSF are often collected. Both qualitative and quantitative methods are used to assess the tubes and differentiate between potential causes. Some examples of qualitative methods might be a visual comparison of the amount of blood present between the tubes, or a comparison of the appearance of CSF pre- and post-centrifugation. So an example of how you can perform a visual comparison between tubes might be to visually compare examples A and C. Example A represents a clear colorless sample, so normal CSF, while example C represents what is called a xanthochromic sample, where red blood cells actually degrade in the CSF, or the color from the breakdown of the red blood cells, or really the hemoglobin within the red blood cells, is converted into bilirubin, which is a byproduct of the hemoglobin, which gives the sample its yellow color, indicating the likelihood that this CSF is a product of an old hemorrhage. In example D, we see the red coloration in tube 1 progressively fade away as more CSF is collected through tube 3, indicating the likelihood of a traumatic tap. But the lab may also look at the color of the supernatant post-centrifugation. If the sample appears colorless, the presence of blood is likely due to a traumatic tap. But if the supernatant appears pink, red, or xanthochromic, a hemorrhage should be suspected. And then, of course, an example of a quantitative method is a cell count, which we'll get much deeper into in later slides.